In this video tutorial, I am going to discuss about triacylglycerol synthesis. Sometimes this is loosely referred to as lipogenesis or esterification. However, remember that neither of these terms is truly synonymous with TAG synthesis. The true meaning of lipogenesis is the synthesis of fatty acids, whereas esterification refers to the process of attaching fatty acids to glycerol through ester linkage. Now, what is the location of this pathway? Triacylglycerol synthesis occurs in almost all tissues of the body. However, it predominantly occurs at three locations: the liver, adipose tissue, and intestinal mucosal cells. Synthesis in the intestinal mucosal cells was already covered in the previous video on digestion and absorption of lipids. So today I will focus on liver and adipose tissue only. Inside the cell, this pathway occurs predominantly in the endoplasmic reticulum, with only a very minor amount synthesized in the mitochondria. Now let's look at the structure of triacylglycerol. A triacylglycerol molecule has one glycerol backbone, which I will represent as a vertical box. Glycerol has 3 carbons, numbered 1, 2 and 3. Fatty acid is attached to each of these carbons, which I will represent as a small horizontal box. So these are 3 fatty acids. So this is the schematic structure of triacylglycerol. Now at first glance, synthesis of this triacylglycerol looks simple. It seems that glycerol and 3 free fatty acids bind directly to give rise to triacylglycerol molecule. But the body doesn't work with the plain, free form of these building blocks. That's where the real story begins, and here is the catch. The synthesis does not use free glycerol. It uses an activated form of glycerol, that is glycerol 3-phosphate. And it does not use plain free fatty acids either. It uses an activated form of fatty acid, that is fatty acyl coenzyme A. So before triacylglycerol can be built, the body has to generate these 2 activated molecules. Let's tackle glycerol 3-phosphate first. There are 3 possible pathways that can generate glycerol 3-phosphate. The first pathway uses glucose. When glucose undergoes glycolysis, it produces an important intermediate called dihydroxyacetone phosphate, or DHAP for short. DHAP can be reduced to glycerol 3-phosphate by the enzyme glycerol 3-phosphate dehydrogenase. This reaction converts NADH plus H plus into NAD plus. This reaction is technically reversible. That's why I am drawing a double-headed arrow. But when glycolysis is actively running, there is a large inflow of DHAP and the reaction gets pushed towards the glycerol 3-phosphate. This pathway occurs in both liver and adipose tissue cells. So let's consider this rectangle as a liver or adipose tissue cell. Now glucose entry into liver cells and adipose cells is not handled the same way. In the liver, glucose transport happens through transporter called a GLUT2, and GLUT2 does not depend on insulin. So glucose entry into liver cells depend directly on the plasma glucose concentration. In adipose tissue, glucose transport happens through GLUT4, and GLUT4 is entirely insulin dependent. Without insulin, GLUT4 will not let glucose into the cell at all. Now let's talk about second pathway for synthesis of glycerol 3-phosphate. This pathway also occurs in both liver and adipose tissue cells. So I will show it over here. This second pathway starts from pyruvate or oxaloacetate, which can go through multiple steps of gluconeogenesis to form dihydroxyacetone phosphate. And once we have DHAP, we already know the rest of the story. It converts to glycerol 3-phosphate. The third pathway is the most direct one. It occurs in liver cell but not in adipose tissue. So let's say this rectangle represents liver cell. In this, glycerol is directly converted to glycerol 3-phosphate. This phosphate comes from ATP. So simultaneously ATP gets converted to ADP, and this entire reaction is catalyzed by the enzyme glycerol kinase. You may ask why adipocytes cannot utilize this pathway. The answer is that the enzyme glycerol kinase is absent in the adipose tissue cells. But here, don't misunderstand that adipose tissue cannot synthesize glycerol 3-phosphate. It can very well synthesize glycerol 3-phosphate by utilizing first and second pathway, but not by this third pathway which utilizes glycerol directly. Now let's activate the other building block, the free fatty acid. In the blood, there is a free fatty acid, but calling them free is a bit of misnomer. In plasma, they are actually bound to albumin. One molecule of albumin can carry up to 10 molecules of free fatty acid. As this albumin fatty acid complex approaches a cell, The fatty acid dissociates from the albumin and is picked up by a specific membrane transporter called the fatty acid transport protein. Once inside the cell, it stays bound to a fatty acid binding protein or FABP. This free fatty acid then binds with coenzyme A to form fatty acyl-CoA. The bond formed is a thioester linkage, and this step is not free, it costs energy. ATP is converted to AMP and pyrophosphate, which means two high energy bonds are used up in this single activation step. This entire reaction is catalyzed by the enzyme fatty acyl-CoA synthetase, which is also known as a thiokinase. This activation reaction happens in almost all the tissues of the body, not just liver and adipose tissue. Now that we have both activated building blocks, glycerol 3-phosphate and fatty acyl-CoA, we are finally ready to discuss synthesis of triacylglycerol. Remember, as told earlier, this occurs in almost all tissues and the predominant organs are liver and adipose tissue. We start with glycerol 3-phosphate, glycerol with a phosphate group on third carbon. Now in first reaction, fatty acyl-CoA comes and CoA part gets released. Remaining fatty acid bind at a first carbon through ester bond. So the resultant compound looks like this. The name of this compound is 1-monoacylglycerol 3-phosphate, also known as a lysophosphatidic acid. This step is catalyzed by the enzyme fatty acyl transferase. In next step, a second fatty acyl-CoA adds its fatty acid onto carbon 2 with release of coenzyme A. So the resulting molecule look like this. The name of this compound is 1,2-diacylglycerol 3-phosphate, more commonly called as phosphatidic acid. This reaction is also catalyzed by fatty acyl transferase. Now we need to add a third fatty acid onto carbon 3, but carbon 3 is still occupied by the phosphate group. So we have to remove phosphate group from here. This occurs by addition of water and the phosphate is removed by the enzyme phosphatase. The resultant compound looked like this. And the name of this compound is 1,2-diacylglycerol. With carbon 3 now free, the third fatty acyl-CoA adds its fatty acid there, coenzyme A is released one final time and the resultant compound looks like this. And it is our triacylglycerol. This last step is also catalyzed by fatty acyl transferase. So to summarize the sequence, glycerol 3-phosphate gets fatty acids added at carbon 1 and 2, then loses its phosphate at carbon 3 and then gets a third fatty acid giving the final triacylglycerol molecule. As mentioned earlier, this triacylglycerol gets synthesized predominantly in liver and adipose tissue, but each of these tissue handles triacylglycerol completely differently, and this is where the story becomes genuinely interesting. What is the fate of the synthesized triacylglycerol in adipose tissue? There are two types of adipose tissue, white adipose tissue and brown adipose tissue. In white adipose tissue, triacylglycerol is simply stored and it functions as a major fuel reserve of the body. Here it remains ready for mobilization whenever there is a period of fasting or inadequate food intake. On the other hand, in brown adipose tissue, triacylglycerol gets stored but in very less amount. The unique thing about brown adipose tissue is that it contains a very high number of mitochondria and each mitochondria contains a very high amount of cytochromes, which is actually responsible for brown color of this tissue. In this brown adipose tissue, triacylglycerol is rapidly broken down, releasing free fatty acid that enters mitochondria and undergo beta oxidation to release energy. Here is the very important concept. In brown fat mitochondria, the energy released during oxidation is not efficiently captured as ATP. Instead, it is released directly as a heat. In newborns, this heat generation is critical for maintaining body temperature. And in some adults who retain significant amount of brown fat, this explains why they can eat substantial amount of food and still stay lean. Their fatty acids are literally being burned off as a heat rather than stored. Now, what is the fate of synthesized triacylglycerol in liver? Interestingly, the liver actually synthesizes even more triacylglycerol than adipose tissue does, but it handles it completely differently. Very little stays stored inside the liver. Most of it is exported as a nascent VLDL into the blood, where it matures into VLDL and it functions to deliver endogenously produced lipids to the peripheral tissues. Finally, let's connect this to whole body regulation. In a well-fed, adequate nourished state, glucose inflow into the blood increases, which triggers insulin release from the pancreas. Because adipocyte glucose entry depends on insulin, through GLUT4, glucose entry into adipocytes rises. This raises a glycerol 3-phosphate level, which allow more free fatty acid to be esterified into triacylglycerol, and as a result, serum free fatty acid level actually go down. In starvation, the opposite happens. Decreased glucose inflow leads to decreased insulin secretion. And in diabetes mellitus, there is either an absolute deficiency of insulin or the insulin resistance is there. So functionally, insulin action is also decreased. In both situations, less glucose enters in adipocyte, so less glycerol 3-phosphate, so less utilization of free fatty acid for triacylglycerol synthesis, and so serum free fatty acid level rises. So we can say that adipocyte metabolism is the main determinant of serum free fatty acid level, and serum free fatty acid levels have a profound effect on the metabolism of essentially every other tissue in the body. We can say that the adipocyte's influence extends well beyond adipose tissue itself. So to bring it all together, triacylglycerol synthesis needs two activated building blocks, glycerol 3-phosphate and fatty acyl-CoA. This combines through enzyme catalyzed steps to form the final triacylglycerol molecule, which the liver exports and adipose tissue stores or burns for a heat, all tightly regulated by insulin status. So that's all for this video. In the next video, I will discuss about triacylglycerol lysis. If you have any query or confusion, please write it down in the comment section below. Thank you.